Assalamu alaikum. Dr. Yasir, can you hear me?
जी वालेकुम जी डॉक्टर साहब वी कैन हियर यू
Thank you very much. Sorry for the late. Actually, technology just at the moment when I was going to start, it just I don't know what happened, so I have to reinstall it. Anyway, so should we start now?
Yes, sir, I will officially start the event. I am the host.
Okay.
So,
यस यस कदर साहब आई कैन हियर यू इट्स ओके Mahnur is our biggest student and she is going to coordinate it.
Okay. Okay. All right. Thank you. Thank you very. Okay. One more thing. Should we record this? I mean, who is the administrator? Yeah. I mean, कौन इसको जो है ना वो कर रहा है?
अस्सलाम वालेकुम डॉक्टर साहब कादिर साहब
जी
मैं
जैद
जैद
साहब। जी जैद
जी कैसे हैं
कैसे हैं आप? ठीक।
अल्लाह शुक्र कादिर साहब
ठीक
मैं
ठाक।
रिकॉर्ड कर रहा हूँ इसको और ये यूट्यूब पे भी लाइव है तो उस पे भी रिकॉर्डिंग हो रही होगी इसकी
Okay. All right.
ओके
So I can share my slides from here. All right.
जी जी आप करें स्लाइड शेयर प्लीज
Okay. All right.
okay, so good evening and assalamu alaikum, everyone. I am Mahnur Khan, the president of BioWare Society. I welcome you all to today's webinar in Nanomaterials as Antimicrobial Agents, Tiny Particles, Big Effects. This webinar is brought to you by the Department of Life Sciences in collaboration with UMT BioWare Society. Before beginning, let me apprise you all about the question and answer session, which will be held at the end. Kindly be patient and type your questions in the live chat option. We will also be providing e-certificates to students, so you will be asked to comment your name in the chat. Note that the certificates will only be provided to students who attend the webinar till the end. So without further ado, let's proceed with the introduction of today's speaker. Dr. Abdul Qadir Gilani is former European Union Marie Curie Fellow. He accomplished his doctoral degree in nanoscience and technology from Austria. He carried out his research work in Fraunhofer Institute of Biomedical Engineering, Sulzbach, Germany. His work focused on nanomaterials applications, nanotoxicity, and microchip-based imaging systems for biomedical applications. Currently, he's working as assistant professor in the Department of Allied Health Sciences, Superior University, Lahore. So let's begin with the webinar. Over to you, sir.
Thank you very much, Manu, for the introduction. So uh, let's start, I think, then. Okay. I'm going to share my screen. Okay. All right. So everybody can see it, please.
Yes, sir.
So, all right. So it is on the screen then. Okay. Manu, how many students we are having here? Mm, okay. All right. It's not support. Okay. Anyway, so we are actually today we are going to talk about nanotechnology, nanomaterials, and nanomaterials role as antimicrobial agents. So actually these what makes these tiny particles to show some that big effects actually. We will talk about a little bit about the what is the nanotechnology, why the nanomaterials is so special when they are in the nanoscale, and what are actually when these particles are in the on the nanoscale size, what the magic happen on that scale. Okay, so uh, first thing that what is the nanotechnology? So the nanotechnology is actually it's it's kind of combination of actually different uh, disciplines in which it is the creation of the useful functional materials, devices, or systems of any useful size. So focus on that, that the creation of the useful functional materials, devices, and systems. So the three things we are talking about here: creation of useful materials, devices, or the system of any useful size through the control manipulation of the matter on the nanometer length scale. So let's stop here. Uh, it basically nanotechnology is controlling the matter or manipulation the matter at nanoscale matter. When the any matter which is at the nanoscale size, it has its own unique properties. So the manipulation of that material on the nanoscale, where we create the materials, or with the combination of that materials results in the devices or set of a system of any useful size, is called the nanotechnology. Where if you further go into the exploitation of the novel phenomena and the properties which arise because of the nanometer length scale. So that is nanoscale, or the research and the technology development aim to understand and control matter at the dimensions approximately 1 to 100 nanometer scale. So that is called the nanotechnology. But the basic thing which I want to convey here is the nanotechnology is about the manipulation or the controlling of any material at the nanometer length scale. So why we are focusing on the nanometer length scale? Because again I'm saying that the properties of the material when they are on the bulk size and when they are on the nanometer scale size, it's totally different. So why it becomes different, we will be discussing further. So the one of the part of the nanotechnology is the nanomaterials. So basically, why the international standard organization defines the nanomaterial as objects are classified as the nanomaterials if one or more dimensions in the size range of 1 to 100 nanometer. For example, I say that I have a cube, so it has three dimensions: its width, height, and length. So here is, if I can, yeah, this is kind of. So in that object, any of that three dimensions, where it's a height, length, or the width, is in the dimension or the size range of 1 nanometer to 100 nanometer. We will call it the nanomaterial, or any other object of any shape. I mean, so whether it is round or whether it is rod shaped or sheet or the darts, if any of the dimension is in the range of 1 to 100 nanometer, is then we can call it as nanomaterial. For example, here if it is on the in the round shape, we will call it the nanoparticles. If it is in the cube shape, we'll call it nanocubes. If it is rod, then nanorod. If it is like sheet here, that is the graphene sheet, that we will call it like nano sheet or the nanodots. And there are some kind of the shapes you can see under these shapes. These are basically the actual nanoparticles, and these are the electron microscopic graph of these nanomaterials. So please, if between that, if you have any question, you can ask me about because I have no idea how much you know about the nanotechnology or the nanoparticles. So that's why I included a brief introduction into that. Okay, so then basically now we will try to understand what is the nanometer length scale actually. So what does that mean? What the size actually we are talking about? We need to know that first, which before going into the details in the application of the nanomaterials or in other aspects of the nanomaterials. For example, if you start uh, from the bottom of that, here we have the one carbon atom which is size, its size is about 0.16 nanometer. The one carbon atom size is 1.1 nanometer. Or if you have a single if you have a DNA strand, its size is about uh, 2.5 nanometer. Or if you take that is the single wall carbon nanotube as a chemical sensor, that is its size is 2 nanometer scale, 2 nanometer. And if you go further, a molecular gear can be range from 10 to 100 nanometer, quantum dots, 300. Could anybody tell me that how many nanometer we have in one microns? Okay, we have 1,000 nanometer in 1 micron. So if here we have a red blood cell, which is of 8 microns. So I hope that with that, the comparison which we draw from the bottom and coming to that red blood cell, or if we have the dissect of the human hair, that can the diameter of the dissection of the human hair can be the size of the 50 to 100 microns, depends. So that's what I'm talking about. That is the nanometer length scale. So we are talking about very actually small or the tiny particles. Okay, so when the materials are on the nanometer length scale, why they become so special? It's actually their volume to surface area ratio. When they are on the nanometer scale, the availability of the surface area is very high as compared to the volume. It is something like that. For example, if you have a uh, mm, mm, for example, we have a one bacterial cell of you can 1 micron size, and the other bacterial cell is uh, 2 micron size. So the 1 micron size cell, bacterial cell, have the more surface area to grab its food or minerals or whatever, as compared to 2 microns, because the 2 micron bacterial cell have the more volume as compared to the 1 microns, but the surface area of the 1 micron is more as compared to the 2 microns. Yeah. So that's why it is the same with the nanomaterials. When they are as they will be small, they would be having the more surface area, and that's actually that surface area make them unique with the other bulk material, because so they have the more reactivity. Or in the other sense, we can say it like that, that the interface of the atoms or the electrons or the reactivity, we can say that when they are on that scale, it is much higher as compared when the material is on the high on the bulk scale. So that's why they become so special at this nanometer scale, and their properties are changed. Okay, so now come to the that that is kind of the application. That is the one of the application of the nanomaterials as the antimicrobial agents. But first we have to I will discuss some terms here, like the antibiotics and any natural substance produced by various species of the microorganisms, which include bacteria, fungi, actinomycetes, that has the ability to kill the microorganisms or the pathogens, here we could say. And then the antimicrobial agents that include the synthetic, I mean that they are synthesized or they are produced chemically in the library, or the naturally obtained drugs. For example, the penicillin from the fungus, naturally obtained drugs that attenuate the microorganisms. So these kind of the drugs are called as the antimicrobial drugs. Another term which is of very hard debate these days, the antimicrobial resistance. That occurs when the bacteria, viruses, fungi, and the parasites change over the time and no longer respond to the medicines, making infections harder to treat and increase the risk of disease spread. So, I mean, the change, the bacteria or the virus change the strategies. So, to minimize the effect of the antimicrobial agents or the antibiotics. So, and they are causing these kind of the bacteria or the viruses are these days causing the severe infections throughout the world. Drug-resistant bacteria lead to the poor clinical outcomes, increasing the healthcare cost and mortality. Definitely, we when the number and the intensity of the antimicrobial resistance increase, we have to invest more money in, and then it results in the higher cost, and also that death rates also goes high due to these. So these are the basically actually the risks we are facing. These both gram positive and gram negative bacteria which display resistance to the multiple drugs, which is a serious concern in the healthcare mm, circle. And the other the strategy which the microorganisms follow, like the biofilm formation, which complicate the treatment of the various infections. And when the bacteria or the multiple microorganisms are in the form of the biofilm, it becomes more harder to get rid of them. So, according to the U.S. Center of Disease Control and the Prevention (CDC), which is their central body in the U.S., which leads the government about the coming concerns in the healthcare area, that more than 2 million antibiotic resistant infections occur every year in US and cause 23,000 deaths, which is very high. And if the 2 million antibiotic resistant infections are happening in the world, most you can say the good healthcare system. So it means that in the other developing countries like Pakistan or India or the Bangladesh, there we are facing a much bigger problem than there. So antimicrobial drug discovery is not economically attractive, as we know that the governments and the private companies invest a lot uh, money on discovering the antimicrobial drugs, mm, and sometimes it is successful, sometimes it is not successful. So there is always a big risk. The investor also take cautions while they are investing in these areas. And the other hectic thing is that once the drug is discovered, then it comes the regulatory requirements, which is also the challenging. You have to go through a very difficult laws, and you have to inline yourself with these laws to prove these kind of drugs. So it is also a very hectic thing to discover a new antimicrobial drug. So that's why it is the need of the hour that we just not focus on the novel bacterial agents, which is we have to look for the new strategies. The emerging nanoparticle based material strategy in the field of antimicrobial therapy could be a very attractive strategy to counter these that health concern due to that antimicrobial resistance bacteria. Interest in the use of these materials stem from the morphological and the physicochemical properties such as high surface area to volume ratio, physical and the chemical property which has been successfully used for the other applications. Nanomaterials have been used much in many other areas, so and successfully they have shown their application. So that is so that and now it is also investigated a lot by the scientists to look for these new strategies against these multi drug resistance bacteria. So why it is important? And the bacteria applications of the nanotechnology are gaining importance to prevent the catastrophic consequences of the antibiotic resistance. The nanotechnology can be implemented as preventives, diagnostics, drug carrier, and the synergetics in the antibacterial therapies. So there are the different strategies in the microbial nanotechnology. We can, for example, we can use nanotechnology for the preventive measures. For we can diagnose easily and cost effectively the infections. We can use them as these antibiotics with combination the nanoparticles, which can be more effective, and also they can also help to understand the whole system of the antibacterial therapies. So these are the different strategies we can put through the microbial nanotechnology to fight these infections. The unique properties of the nanomaterials compared to its bulk form make them favorable for antibacterial therapies. Okay, G. There are so we talk now about now the factors which actually affect the antimicrobial activity of the nanoparticle. The first and the very important thing is the charge, the surface charge on the nanomaterials. Surface charge means that whether they are having the positive or the negative charge, you can say. The varying the surface charge in the nanoparticles in a marked function of the antimicrobial activity occurs since the bacterial surface shows slightly negative charge. As we know that the bacteria or our eukaryotic cells, they have this the negative charge on the cell membrane. So it becomes then very important that what kind of the surface charge our nanoparticles should have to attract these the pathogens. Size of the nanoparticle, that is again of very important property of the nanomaterials. For example, if you have this very small nanoparticle size, thing is that we are saying that the nanoparticle, or nanomaterial is that thing which is in the range, whose one dimension is in the range of 1 to 100 nanomaterial. So it means that we have the range. So as we have the low size, between that range, we have the more effective antimicrobial activity of these nanomaterials. Shape of the nanomaterial, again, then it comes shape of the nanomaterial. That is also the very important. For example, whether it is of cube shape, whether it is of rod shape, whether it is a sheet, or globular. So we have to find that what kind. First, it is we have to look what kind of the bacteria we are going to fight, whether it is gram positive, whether it is gram negative. Then we have to see the structure of that cell membrane. Then we have to strategize it, that how we can design our nanoparticles which are more effective as compared to the other shapes of the nanoparticles. So then it comes to the concentration of the nanoparticles. For example, what concentration we are going to use against the or what is optimal concentration of the nanoparticles that we can use against these pathogens. For example, normally a bit high concentration is better, but it is not exactly the right thing. Sometimes, for example, if we are using 25 microgram per milliliter concentration of the nanoparticle, it can be more effective as compared to the 50 microgram per ml concentration of the specific nanomaterial. So we have to optimize these conditions, these factors, which is in our lab, which include charge, size, shape, and the concentration of the nanoparticles. So we have to take care of these things before we are going to use the nanoparticles in that. So what is actually the mode of action uh, uh, of these nanoparticles, how they are going to kill the pathogens? So they different things. The main part of that, the mode of action against the pathogens of the nanoparticle, first is the physical interaction, then is the release of the ions and the production of reactive oxygen species. So these are the, you can, in the broader aspect, we can say, and each and every we would be discussing in the coming slides, that that how the different nanoparticles are using these kind of strategies to fight against these the nanowire. For example, physical interaction means that when the metal, normally we use the metals or the nanoparticles, which are metals or metal oxide, against these nanoparticles, against these pathogens. So for example, when nanoparticles are coming in the when interacting with the with the bacterial cell membrane, so they can disrupt or they inhibit their electron transport chain. वो उसको रोक सकते हैं, वो उसको जो है वो जो इलेक्ट्रॉन ट्रांसपोर्ट चेन को जो है वो डिस्टर्ब कर सकते हैं. अच्छा, देन जो है वो they can inhibit the protein synthesis of the nanomaterial, protein synthesis by the cell in the cells. So the other thing is the release of the ions. There is a phenomena which is called the dissolution. Then the metals is actually, its dissolution is kind of, you can say, the self-destruction. When the metal or metal oxides are interacting with the cells or the pathogens, they release ions. These ions are taken up by the cells. They can interact, or they can go to the nucleic acid, and it can cause the nucleic acid degradation, inhibit the electron transport chain, or they can also produce the reactive oxygen species. What is the reactive oxygen, Shivi? They are actually, it is the oxidative stress, actually, these the ROS cause in the cell. Oxidative stress can result in the sudden death of the cell, which by degenerating the nucleic acid, by prohibiting the mitochondrial or other different cell organelles functions. So and it can attenuate or disrupt the cell functions. If anybody have any question until now, you can ask me. Okay. Okay. These are the actually we have here the kind of the detail here of the that how it is going to um, how nanoparticles actually act. The ROS or the reactive oxygen species, it is when the nanoparticles are interacting, they can produce the superoxide anions, hydroxyl radical, hydrogen peroxide are produced after the exposure to the nanomaterials such as the metal oxides. They these actually, as I earlier said, that they can cause severe cell damage in the form of DNA degeneration, and which can subsequently result in the automatically in the cell death. By binding to the bacterial cell wall, it can compromise the cell membrane integrity, which and can break the cell membrane, which can result in the leakage of the cytoplasmic material from the cell, and of course, when the cell membrane is compromised, it is going to die soon. Okay, but the physical damage, as I told that, as I tell you, that physical damage can be the cell membrane, or it can change the shape of the cell. So that is, and then the next is the interruption of the electron transport protein, oxidation, and the membrane column. Positive charge of the nanoparticles is critical for the antimicrobial activity since the bacterial cell membrane is negatively charged. So it is very important here. I was going to talk about that because the bacterial cell membrane is having the negative charge. So to make the nanoparticles more effective against the bacteria, we have to always have the positive charge on our nanoparticles. If we are having the positive charge, then and that the pathogen cell membrane can attract the nanomaterials. If we are having already negative charge on this on our nanomaterials, then it is going to the cell membrane is not gonna attract. It will repel the nanomaterials, and our nanomaterials will not come in. Will not be interacting with that with these pathogens. So while they are not interacting with the pathogens, then how they can kill these the pathogens? So that is what we have to take in the consideration. So now we come to the certain kind of the the particles which are actually uh, um, which have proved their strength against the against the pathogen. The silver nanoparticles are the most and are the top, and from the start of the human history, we are using silver in many things. But these nanoparticles are used at the apparels, footwears. For example, I would example it like our socks. Their internal lining is or the coating is with the silver nanoparticles. As we know that the socks in the shoes produce a very they are very smelly. Our foods are very smelly. These are the bacteria actually. When we have the lining of silver nanoparticles on these socks, they are not gonna produce that bad odor from our shoes and from our socks. In the paints, in special surfaces, where we need to have the we don't want the bacteria. These kind of the paints are done there, which have the silver nanoparticles. When the silver particles are on the surface, there is less chance of the contamination. Wound dressings, especially the burns in cause of the burns on our skins, the dressings, they are coated with the silver nanoparticles. So these silver nanoparticles are actually functioning as antibacterial agent in these dressings. In the appliance, in the cosmetics, in the plastics, the nanoparticles are used in these different as an antibacterial agent. So how actually they are going to the work as the antimicrobial and silver nanoparticles. The, Normally the silver nanoparticles having this strong surface positive charge are attracted by the bacterial cell surface. So when they are attracted, they are attached to the bacterial cell membrane. They are going to destabilize the bacterial cell membrane. They induce the induction of the toxicity and the oxidative stress by the generation of the reactive oxygen species, and the modulation of the single instruction as the electron transport chain. Again, this is these the four strategies are applied by these silver nanoparticles against the pathogens. So here we say that as uh, in that picture that and that silver nanoparticles are actually producing the oxidative stress, which later results in the DNA destruction, and if and also the destabilization of the cell membrane. So once the basic thing is that the silver nanoparticles do when they are interacting with the cell membrane, they are actually compromising the cell membrane integrity. So they are actually, you can say that they are quite capable of making the holes in the cell membrane. When they are making when there is a damage or the cell health is compromised, it's definitely it will uh, produce the oxidative stress, and then that oxidative stress will cause the DNA defragmentation. So that's how these pathogens are um, killed through the mechanism of the silver nanoparticles. Silver ions can interact with the DNA strand, forming the complexes with the nucleic acid between the purine and the pyrimidine bases, and disrupt the hydrogen bonds. If the hydrogen bonds is disrupted, that will result definitely in the DNA defragmentation. Okay, ji. After silver, we came to the titanium oxide. Titanium oxide is also very has been reported definitely against very drug-resistant bacteria. It is long been known for its ability to kill both the gram-positive and the negative, which is a big property of that titanium oxide. Uh, More recent reports have shown that its efficiency against the various viral species and the parasite. So it is very, it has very broad antibacterial or an antimicrobial spectrum. It is effective against the gram-positive, effective against the negative, also against the viruses and the parasites. Titanium oxide dioxide is effective against many bacteria, including the spores of the. You know that the spores are very difficult to deal with them. So they are. It is reported that they are very effective against the spores of the bacillus, which is the most resistant organisms as known. So, and also in that titanium with the combination of silver is used as a very effective antibacterial agent in different experiments. Okay, ji. Copper oxide. Copper oxide nanoparticles exerts their antibacterial activity by the membrane disruption and the ROS production. Copper oxide nanoparticles have been shown to be effective against the various bacterial pathogens. Their antibacterial efficacy is somewhat inferior to that of the silver and the zinc oxide. It is reported definitely that it is mainly effective as membrane involved in the membrane disruption and also the production of the ROS. But as compared to the zinc oxide and the silver nanoparticles, they are less effective. But if we increase the concentration of these copper oxide nanoparticles, we can get the same results. But the thing is that we have to take care take care of that if we increase the concentration, it cannot have any other consequences on the human health. So we have to take care of these things too. Then come to the zinc oxide. Zinc oxide is a very lethal particle for the eukaryotic and the bacterial cells. It is especially it comes with the phenomena of the dissolution. It produces the ions when they are interacting in the media with the cells. Zinc oxide goes to the self destruction and it produce the ions. These ions interrupt with the electron transport chain. They cause the reactive oxygen species, and in the end, that they go down to the nucleus and interact with the DNA and damage the DNA, and it can kill. It has the ability because I myself have observed that within 5 minutes, these zinc oxides are and nanoparticles are quite effective. Just by after the exposure of 5 minutes, the cells were found to be dead. So these are the zinc oxide nanoparticles, and they are found very effective against very drug resistant bacteria like methicillin sensitive staph aureus, vaccine resistant staph MRSA, generally known as these kind of staphylococcus mutants, lactobacillus, salmonella, these kind of bugs, they have been found very effective against these pathogens. So, supermagnetic iron oxide. Basically, these are mostly due to their magnetic nano property. They are used to destroy the biofilms because due to that magnetic effect, we can use these kind of the iron oxide nanoparticles to penetrate by applying the magnetic field that they can penetrate into the biofilm, and then they can disrupt the cell membrane and also the different cell organelles. So that's where they are effective. Then it's again it comes a phenomena of the superbugs. These are the multi-resistant superbugs have emerged due to the abuse of the antibiotics. As you know that especially in Pakistan, there is a severe abuse of the antibiotics. most of the time we don't need the antibiotics, but we are given the antibiotics. So by that use, ineffective use of the antibiotics, the emergence of that multi-resistant bacteria happens. So they, for example, Mycobacterium tuberculosis, Enterobacter coliaceae, Klebsiella pneumonia, Staphylococcus aureus. These are the Pseudomonas aeruginosa. These are the superbugs actually, and we need to have better strategy against these kind of the multi-resistant superbugs. So there is a study I want to share with you that with the combination of two drugs and the selenium nanoparticles they use against these kind of the pathogens, and the results are quite effective. That what they did is that they used the two drugs, quinine and arginine, and these if you look at that, this graph here, so there are E. coli and Staph aureus. These are the two bacteria. They expose. There are three strategies. Two separate drugs with the selenium nanoparticles, and the both the drugs with the selenium nanoparticles. They use against both E. coli and Staph aureus. So if you look at the blue lines, which is showing that selenium nanoparticles with the both drugs, they show the maximum effect. If you see that this is the exposure time of and if you see that the viability of these bacteria is decreasing drastically within the 60 minutes. If you see that it is with the passage of time, the viability of these superbugs is actually decreasing. So that is as I earlier I said that these drugs were not that effective separately uh, against these superbugs, but when they use them with combination of the selenium nanoparticles, they found a very effective results against these multi-drug resistance bacteria. Okay, so that is the same mechanism that they came in contact with the cell membrane. They cause the leakage of the cytoplasm and allowing the nanocomposites to subsequently invade the bacterial cells and disrupt the DNA structure. Again, there was another strategy. They used the graphene oxide sheets against the E. coli, Pseudomonas aeruginosa, and Staph aureus, and they found that they produce the graphene powder, which were having these graphene oxide sheets, and then they took isolate the culture of the superbugs, and they mix it, and they look for the antibacterial effectiveness of these graphene. Actually, what did if you here see that what the graphene did, it entrapped the superbugs in these sheets, and it compromised their cell membrane, and subsequently it goes down to the DNA, and it disrupts the DNA of these superbugs. and that's how they are found very effective against multi-drug resistant bacteria. So what we have discussed until now is, and it has the advantages in that administration of the antimicrobial agents using nanoparticles can increase the overall ph- uh, pharmacokinetics by progressing the therapeutic index, extending drug circulation, and providing the controlled drug release. As I said before, and we have seen through that graph that when we are using these drugs and in combination with the nanoparticles, they actually change the pharmacokinetics of the effectiveness of these drugs and the nanoparticles. In combination, they are more effective. They can disrupt the pathogen cell membrane and the DNA uh, quickly, as compared if we use them uh, in separate ways. The multiple mechanism of action can be provided by the antimicrobial nanoparticles, which prevent the development of antibacterial resistance by many pathogenic bacteria. Several routes. There is another advantage of the nanoparticles, because that the administration routes, which can include the oral, nasal, parenteral, intraocular, and these are the different routes which can be opted by using that nanoparticles. Nanoparticles makes difficult for bacteria to develop the resistance mechanism against these nanoparticles because of the certain properties of the nanoparticles. So it's not easy for the bacteria to develop the resistance against the nanoparticles. There are also some disadvantages, and definitely it comes when we are using the metal-based nanoparticles. For example, if we are using with the human with against the bacterial cells. So if we distribute these nanoparticles, they will also come in contact with the human cells. And there is the problem of there is a problem that where after their work, where these nanoparticles, the metal particles, will go. So and most of the times it is reported that the metal that the metal nanoparticles remains in the vital parts of the body. For example, it can end up into the liver, it can end up in the kidney, it can end up in the brains where they will not be discharged. So we have to find the ways that and if these metal-based nanoparticles are staying in the body for the long time, they can also produce the ROS, the reactive oxygen species, and the oxidative stress or the inflammatory response within the humans, which can cause the pulmonary diseases, liver degeneration, nephrotoxicity, or the neurotoxicity. So, with the passage of the time, if these metal-based nanoparticles are inside the body, our body is going to react it. So we have to find the or optimize the ways that how we are going to use these nanoparticles. What should be the concentration of the nanoparticles? So we would conclude today's our lecture. Due to the well-described antimicrobial activity against the gram-positive and the gram-negative bacteria, these particles have been proposed as alternative over traditional antibiotics to overcome the bacteria resistance. definitely, as that is one of the alternative strategy, because we are our antibiotics are compromised, and superbugs are reported. So definitely these nanoparticles can be the alternative of that traditional antibiotics. But as we discussed a few seconds ago, the potential risk of the toxicological events in humans when using metal nanoparticles also exist. What we need to do, it's not I'm saying that we cannot use them as the nanoparticles. We have to do the physiochemical characterization of these nanoparticles, because using extensively when we use when we characterize these nanoparticles in all the system ways where we are using. For example, first in the lab, in vitro experiments, just before using the nanoparticles and after using the nanoparticles, we have to characterize them, that how they are behaving, or what are their what about their surface charge, what about their size. And then when we are going down to the animal models, again we have to characterize these nanoparticles before using in the animal models and after using them in the animal models. So understanding of the nanoparticles in vivo behavior during the preclinical and the clinical trial is the vital source of information for the success of the pharmaceutical development. So I hope so that you would like that lecture about the nanoparticles as antimicrobial agent. If you have any question,
Yes,
I
sir.
would
Thank you
really
so much for
like to answer. Thank you.
thank
Thank
you
you.
so much for this informative lecture. We have a lot of questions on YouTube.
Yes.
So I will start by asking the first question. How can
Mhm.
nanotechnology be used in the prevention of diseases?
For example, we can use them especially by making the disinfectants. For example, the best strategy is the not let happen the infection. Okay, so for example, uh, as we say that there are uh, mm, this we can produce the kind of the disinfectants which are the silver nanoparticles or the other nanoparticles base which are highly effective. We can make the materials, for example, in our kitchens and the bathrooms. There are the maximum number of the microbes. Okay, so we can have, for example, we can have the kind of the material on our floor or on what we call that on the surfaces where we use to cut our vegetables, meats, and the things like that. So the thing is that if we prevent these bacteria or the pathogens to stay on these surfaces, we can prevent a lot. And then there are many other strategies. So we can use the packaging of that kind of the packaging, which is of the nanomaterial based, and that packaging of the food materials or any kind of the packaging having some drugs or some water or drinking that that can already prevent you from the bacteria. So in these making the disinfectant, the surfaces that are, for example, in the current situation, a lot of things have been proposed by the scientists that we should have the surfaces in our daily life of use that are the antibacterial or antiviral material. So I think by using that strategies, the nanotechnology can be helpful in the prevention of the infections.
All right, all right.
Next.
Sir, so if we use nanoparticles in hand sanitizers, as you have said that they're very effective against against microorganisms. So, but you also have stated that they possess some kind of harm to humans, like the disease you mentioned. So, how can we control this? Well, like, for example, if we're making a product
Is
based on nanoparticles,
listen.
how
Okay,
will we
good,
prevent
good, good,
the
good. It's
diseases?
very, very good question. The thing is that first I have said that we are already using the silver nanoparticles in the wound dressings. You know, uh, uh, when there are the burns, the dressings on these burns are the wounds, which normally we have the cuts. These kind of the dressing already are having the silver nanoparticles on that. Okay, and it is very commonly used in our hospitals or in our daily life, and they have shown and it is shown no toxic effect yet. But the thing is that when we talk about as antibacterial or antimicrobial agents, for example, as we use them as antibiotics, and when we are taking into our body, right? So here we have to use these nanoparticles after the lot of clinical trials. So the thing is that you have to optimize that what is the concentration is of using these nanoparticles is safe. Okay, or there is another strategy we can use that if we kind of we make the surface of these nanoparticles while using them as antibacterial, or we modify them that they are attractive in our body system and they can be discharged. For example, lipid layering or something, we can play with the nanotechnology a lot. So, the thing is that a lot of characterization, that is that you use both kind of the models in vivo and or in vitro, that when they show no harm, or when they are easy to be discharged from the body, then you can use them, and that's how you can overcome to that safe use of the nanoparticles.
Okay, okay. Sir, you also talked about nanoparticles being effective against many bacterial species, but
Mhm.
are they effective against fungal species and molds?
Oh, of course they are. I mean, there are studies, and I have named in my lecture. If you go back, yes, they are. I think Candida albicans. It is a fungal infection, fungus. It has been shown that these nanoparticles are effective. The thing is that it. Uh, for example, the thing is what strategy you have to you have to find the way that which strategy you can choose to kill these kind of the microorganisms, and or what is the soft target of the, for example, definitely there is a different morphology of the fungus and different morphology of the bacteria. So we have to find what kind of the single nanoparticle or the combination of the nanoparticles or the combination of the drug plus uh, nanoparticle can be effective. So yes, there are studies and which have reported that these nanoparticles, some are not all kind, some of the nanoparticles, that they are effective against the fungal infections. By
Okay.
the way, I think that I can add that there are some ointments or the creams for the skin in fungal infections. They are already in use.
Okay.
And,
Okay, sir. Thank you.
I would suggest you whenever you are using these kind of the creams and definitely the cosmetics. And cosmetics is a very huge industry of the nanoparticles. Your lipsticks contain the nanoparticles. So
Okay, so
I would
very
I would
interesting information.
yes. I mean the most of I would just say that please read the labels on that. They will be labeling that we have used these kind of nanoparticles. Might be they could be the lipid nanoparticles. So I don't. But you have to see. I know them. Yeah.
Okay, okay. Now we have another question. How we use MRI contrast enhancement in nanotechnology?
MRI contrast. I think they. I think these the quantum dots are actually I think are being used in that because the quantum dots are are the nanoparticles which are highly used in these kind of the contrast or live cell imaging or in the kind of the MRI type things. So because why they are so attractive? Because they gives you the very sharp colors. So they can give you a very good contrast as compared to the other things. So
Okay.
yeah.
Yes, we have another question.
Mm,
Can any element of the product table be one of the nanomaterials or not?
no, it's not like that. Yeah, the thing is that you can see. Listen, there is every protein who's who is in the size of uh, in the 1 to 100 nanometer range. It can be a nanoparticle or the nanomaterial. Okay, so come to the definition. Any material, any material. It can be anything on the periodic table, right? If its one dimension is in the range of 1 to 100 nanometer scale, that could be a nanoparticle. But the thing is that the nanomaterial or whether it is whether it has its some application or not, we cannot say that, right? But the thing is that what
Okay.
I'm telling you that the thing is that when the same material is in the bulk size, for example, in the micron size, it won't be having the same properties when it is in the nanometer scale.
All right. All right.
Got it?
Um,
So
yes.
please summarize. Yes, it can be a nanoparticles, but it has its kind of effectiveness or not. We are not sure.
Okay.
Yeah.
We have another question. How can we minimize or control the toxicity effects of nanoparticles
How we
in
can
humans, of course?
in human.
Minimize or control the toxicity effects of nanoparticles in
Listen,
humans.
the project in which I was involved in your union as a Medicare fellow, that was actually about the project where your union is going to regulate uh, the products which are using the nanoparticles or nanomaterials. They want the data from us that of the different aspects that mm, what concentration of the nanomaterial is safe, what type of the nanomaterial is safe, and what is not safe. Okay, for example, as I uh, the the effectiveness or whatever the application of the nanoparticles depends upon its surface charge, its size, its shape, and its concentration. So now we have so we have to regulate it like that, that what is the concentration of the nanoparticle in some specific product, so whether it is safe or not. For example, if it is the concentration is of, for example, in a product is 25 microgram in per gram is safe, but if we increase the concentration, okay, like if you go to the 50 microgram, it might not be safe. Okay, the other thing, uh, whether the surface charge, if you are going to use a nanomaterial, which is coming in direct contact with the human, for example, skin or something like that. What is the surface charge on that? How is going to interact with the skin cells? So we have to then here we have to actually categorize the nanomaterials. We have to do this the extensive physiochemical characterization of the nanoparticles. Then we can regulate it. And by the way, the nanomaterials are not the bad guys actually, really. It's yes, they have their toxic effect can be, but it's not that much until now. So if you take the measures of these things, first the intensive experimentation, extensive physicochemical characterization, you can minimize the toxic effects. But right now, their applications are really very high as compared to the toxic effects. So
All
yes,
right, all
by
right.
taking care of these things, we can say that yes. Uh,
Okay.
by the way,
So,
these batteries these days we are using, or these long hours batteries, these are also the nanomaterial based batteries actually. So there are many positive things regarding the nanomaterials. Yeah. Next, please.
yes. So, we have a plethora of questions on YouTube, and but we're running out of time, unfortunately. So my apologies to the audience whose questions I could not ask you. So I guess we should conclude the session. Thank you so much, sir, for your time. Our students learned a lot from today's webinar, and by the feedback response that we are receiving and the questions we are receiving, we can tell that this was a very informative webinar, and this was a very interesting topic, and everyone is very much interested in this. So thank you for your
Thank
time,
you.
sir. Thank you so much.
Thank you very much. I mean, if due to the time, I cannot answer a lot of, might be many questions, but still it is open. You can write me. You can have my email address through Dr. Yasir's, Dr. Zayed. I mean, I can answer it. I would love to. Thank you very much. Thank you. Allah Hafiz.
Thank you. Allah Hafiz.